Welcome back, guys. We are talking about the eukaryotic DNA replication process. In the previous video, we have talked about the initiation of eukaryotic DNA replication, right? And we are at the phase where S phase is reached during the cell cycle of a eukaryotic cell division. Now, during that S phase, at the beginning of S phase, we have seen three important protein factors acting as the function of DNA helicase and unwinds two different strands from each other, separates two different strands from each other. And those three proteins, one is MCM, this is the MCM protein, remember, MCM. MCM means mini chromosome maintenance factor. This is CDC45, cell division cycle protein 45, and this is the GIN. I forgot the name full form, but these are the three different proteins that are involved and that are acting as a helicase and unwinding two different strands, melting two different DNA strands from each other, and they are moving towards a particular direction where exactly the polymerase will move. So here the directionality of this replication is this is the directionality. So this is the 3' directionality normally of this replication. Now remember, one thing I should say before going into the details of this again, this thing is formed during the initiation, at the initiation later stage of initiation phase. And I will recommend you to go back and watch that initiation video first, otherwise you won't be able to understand that. So once we have this scenario, and I have told you that there are two different parental strands and the DNA replication should occur simultaneously in both the strands. Okay, so if it wants to continue this process in both the strands together, the only way is possible, because you know the polymerase can only migrate from 5' to 3', only from 5' to 3'. This is the directionality of a polymerase for the newly synthesized strand. So let's say this parental strand is 3', this is 5', and let's say this. Okay, so if this is the scenario, we can easily synthesize a new strand from 5' to 3' at this direction. While the replication fork, this is the structure called replication fork, because see, one double stranded DNA, now fork like separation, replication fork, and the fork is moving at this direction and polymerase will move at this direction. It is completely supported, and the strand that will synthesize here will be called as the leading strand, because the directionality of this strand synthesis is same as the directionality of the fork movement. But on the other hand, the other strand, it is opposite, like 3 to 5. So to synthesize 5' to 3', we need to go in this direction. We should go at this direction. So how could we solve this problem? We cannot go into opposite direction, because in this direction there are this DNA segment are already attached, they are not melted. The DNA melting is going on in this forward direction, because this melting complex placed here and moving in this forward direction. So the only way to carry these two processes simultaneously is to make a bend over this other strand, so that as we bend this strand, now you can see we are going from 5' to 3', right, due to this bend, and we are following this bending for prokaryotes as well as for eukaryotes DNA replication. Okay, and this loop is very, very important, the replication loop, very important, and this loop formed in the lagging strand. So the strand we will prepare here will be the lagging strand. It will be fragmentized multiple times. This synthesis will not be continuous. On the other hand, the leading strand synthesis will be continuous. Now once we know this leading and lagging strand differentiation and the loop like structure here, now the second thing what I want to talk about is that this melting complex is moving towards this direction, the right hand side according to you, this direction, my right hand side. So in this case, as it is moving to this direction, what will happen? It will unlock, I mean, as the DNA is melted, that means the hydrogen bonds are broken. So what is happening here? The twist of the DNA, the twist number is decreasing, because we are opening the DNA, the twist number is decreasing. Remember, a concept of DNA linking number equals to twist plus ride. This is the idea, right? LN or linking number, whatever. twist equals twist plus rise. So what we are happening here, this twist number is decreasing due to this melting of the DNA in this forward direction. So whatever DNA segment downstream of this melting complex are going to have less twist number. Okay. So to compensate that, it creates a tension to the DNA. To compensate with that, the DNA increases its ride number to make this linking number constant. So the ride number is now increasing by the DNA in this downstream region. So as the ride is increasing, ride means it is wrapping the DNA upon, DNA is a coiled domain, so DNA is a coiled molecule. Now it is further coiling itself, so it creates a supercoil, a positive supercoil at this downstream region, creates a positive supercoil at that region. So once it is creating the positive supercoil, that means DNA is wrapping around itself, so it forms like a knot. So movement for this melting complex is difficult further downstream due to the knot formation there. It creates enormous tension in the DNA, and to leave that supercoiling and break that supercoiling and to restore the DNA conformation again, we need a specific enzyme called topoisomerase. Topoisomerase release the tension by removing the supercoils. Here the topoisomerase can release this positive supercoiling in this downstream region, so that the melting complex can move and DNA synthesis can go on, right? So that is the idea I want to talk about. Topoisomerase is acting somewhere in the upstream. Let's imagine here, this is topo, it's acting upstream. So this is the complex that we have in the S phase we are in. Now we'll see the actual elongation process and how it works. We've seen the loop formation. Now we need, we all know that the DNA replication or the DNA polymerase can only elongate the 3' hydroxyl, because it cannot start the DNA replication or adding nucleotide from scratch. It is not de novo. RNA polymerase is a de novo polymerase. That means it can start DNA synthesis from RNA synthesis from scratch, but DNA polymerase cannot. So for DNA polymerase, we need stretch of ribose nucleotide act as primer, right? So these are primers, these red sections, these are primers that we require to elongate the stretch by the DNA polymerase. We have primer. Now we need only one primer at the beginning of the leading strand, but we need multiple primer for the lagging strand, because we have segments, discontinuous formation. And after the separation of these two strands, we also need certain proteins which are called single strand binding proteins, because those single strand binding proteins separate those two parent DNA from each other and do not allow them to reanneal with themselves, and also increases the stability of single stranded DNA, because single stranded DNA is less stable than double stranded DNA. So here that stability is maintained by the action of those single strand binding proteins. So we see some of these single strand binding proteins attached here all around place like that. SSBs. There are different names of SSBs out there. So whatever, this is the scenario. After this, in the leading strand, we know the polymerase that will act in the leading strand is polymerase epsilon or epsilon, whatever. On the other hand, the lagging strand synthesis will be made by DNA polymerase delta. Now there is an important feature about the polymerization in this phase, that is, for the first few nucleotide synthesis, none of this delta and epsilon take out this process. Polymerase alpha required for the synthesis of first few stretches of nucleotide of the DNA. These are the RNA primers, and these primers are added by enzyme primase, right, or PRI. This is the enzyme called PRI, primase enzyme. That primase enzyme attach all this RNA primer, but during that time, the first few stretch, because these primers are RNA or ribonucleotide, not DNA sequence, these are RNA sequence. We don't need that. In future, after the completion of this process, we should remove this. We have to remove this. But the thing is, the few first stretch of nucleotide, deoxynucleotide, those nucleotide will be synthesized by polymerase alpha, not the polymerase epsilon or not the polymerase delta, by polymerase alpha. So polymerase alpha and primase acts together. Polymerase alpha and primase acts together. So I am drawing it. This is the polymerase alpha along with primase. They act together. Primase attaches the priming sequence, the RNA sequence, few first stretch. Then this DNA polymerase, this is the DNA polymerase. Remember DNA polymerase alpha. Then DNA polymerase alpha start adding first few stretch of the sequence. I am drawing this with this blue color. First few stretches are made with DNA polymerase, polymerase alpha, right, in both this end. Right. So once that thing is done by polymerase alpha, few stretch 2, 3, 4, 5 nucleotide sequences, then polymerase alpha just leave the place, as well as the primase also leave the place, because this complex DNA polymerase alpha and primase altogether leaves the place, and then the actual polymerase will load. The polymerase who will carry out the whole polymerization process will load. Now in case of leading strand, the polymerase that is loading here is the polymerase epsilon. Right. So let me draw it. So polymerase epsilon will be loaded, and polymerase epsilon will start adding nucleotide sequences. So let's say, let me draw a giant. This is polymerase epsilon, and it is carried with a sequence specific structure called pCNA ring. Okay, the matter I will be talking about later, but this is the polymerase epsilon along with the pCNA ring. They will start adding nucleotide sequences for the leading strand, and it will start adding nucleotide sequences throughout this time without any disruption. That's why it's called leading strand, continuous synthesis. On the other hand, the lagging strand, they will also load another polymerase called, remember, the polymerase delta. Again, synthesis. So here we go, the polymerase delta. It's not delta, it's polymerase delta. And alongside we have pCNA ring. Okay, so polymerase epsilon for the leading strand, polymerase delta for the lagging strand, but both of them have this pCNA ring, right? But remember, the synthesis process different in the lagging strand, because say this polymerase delta will start adding nucleotide sequences, and once it is in direct contact with this primer, with the upcoming primer, the last primer, it will stop the synthesis again. Then again it will go back and start doing again, and actually the synthesis process here starts from this end site, right? So polymerase delta will load with pCNA, start adding nucleotide. Once it is doing this process, it is carrying on this process, and then after all, once it is reached and in contact with the RNA primer, it will fall off, and then again it will initiate it from somewhere back, somewhere from here, then again it's doing, again meeting the RNA primer, falls off. This is how the process works. That's why they require multiple primers, and that's why they terminate so much, right? This polymerase epsilon carries the highest processivity. Polymerase epsilon can add the maximum number of nucleotide sequences at a time. So it is with the highest processivity, and it can add the maximum number of nucleotides without any error and failure. It does some error. It also have the 3' to 5' exonuclease activity, or which is called as the proofreading activity, to replace those misincorporated nucleotides. Polymerase delta on the other hand is also good, but the thing is it is not as much effective as the polymerase epsilon. So delta will go on, and whenever it is in touch with the RNA primer, it will fall. Now here is an important strategy for this polymerase delta in this lagging strand, because remember, lagging strand, once after this replication is done, the polymerization is done, what we'll find? We'll find a strand that is filled with RNA primers in between, RNA sequence in between. So it is a problem. We need to take out all these RNA sequences out and fill it with the DNA sequence, and that is very important. So for that reason, the polymerase delta, once it is adding nucleotide sequences one after another, with the help of PCN ring, it is start adding nucleotide sequences, and whenever it is in contact with this RNA primer, it does not stop then and then. What it does, it start cleaving some of the RNA sequences, 2 to 3 RNA primer sequences. It start cleaving 2 to 3 RNA sequences from there. 2-3 RNA sequence from the primer region. It will cleave that, and then it will recruit another protein called FEN1. This FEN1 is kind of a endonuclease enzyme. Right? This FEN1 will start cleaving the rest of the primer sequence, and then FEN1 will fall down, and then rest of the gap will be filled by delta, polymerase delta. It will fill the rest of the gap. Once they fill the rest of the gap, it will form a nick at the end, and the nick will be sealed by DNA ligase 1. So this is the sequential event that is going on. Remember, we don't bother about the leading strand synthesis, because there is no problem whatsoever with leading strand synthesis. Always there are problems regarding the lagging strand synthesis. So once this once this delta reaches the primer, it will start cleaving 2 to 3 nucleotides, then it will recruit this FEN1, FEN1 cleaves the rest of it, it will just chop off everything of RNA template, it creates a blank region, delta fills that blank region, then it leaves a gap, it leaves a nick, say let's say it is filled, and now delta fills this nick, but there remains, delta fills the gap, but there is a nick. There is a difference between gap and nick. Gap means definitely there is a place for a nucleotide to be placed, so nucleotide gap, but nick means there is no nucleotide left to be attached at there, everything is added, only the phosphodiester bond is broken. So the phosphodiester bond is joined by the DNA ligase 1 to seal that nick, right, called nick trans. So nick sealing here. Okay, so these are the sequential events, remember. Now another thing about the PCNA ring, why we need this PCNA ring? This PCNA ring, they are also called as the clamp loader proteins. Now this clamp loader and beta clamp, the clamp proteins, these are very, very important, because these clamp proteins can hold on to the DNA for longer period of time, because you know the polymerase are complex structure, polymerase epsilon, polymerase delta, they are very complex. So, and they are very weight also, they have a heavy structures, huge molecular weight. So they cannot attach with the DNA for very long period of time. They can eventually fall, they will eventually fall off. So to prevent the premature termination of the DNA polymerization, we need to use this PCNA ring. The PCNA ring acts like a loader, a clamp. It will hold on to the DNA strand, it will not allow the polymerase to fall off pretty quick and start adding multiple number of nucleotide sequences throughout a long time. Okay, that thing is done. That's why the PCNA ring is very, very important. Okay, so in both the way, this is the way of elongation process. And remember another thing, so this that I have told, that this FEN and how this process works. There is another way. Sometimes this FEN proteins and this delta, they have some problem with working together to remove this RNA primer. In those situations, they recruit another protein called DNA2. This is another protein which can do both this job of delta and FEN1 at itself. It will cleave that whole RNA segment apart, right? Now this process of RNA primer removal should also be done in case of leading strand, but only once. Okay. We don't need to remove this blue stretch which is done with the DNA polymerase alpha, because this is DNA segment. We don't need to remove that. The same thing. We need to remove this primer only, and that thing we done in both the strands. Okay. So the small fragments that we prepare, as you can see, the small fragments from primer to the DNA small fragments, from one primer to the end of another primer, end of one primer to the start of another primer. These small fragments are called Okazaki fragments, and these fragments lengthen kind of 300, 400, even up to 1000 base pairs in case of eukaryotic DNA, while in prokaryotes they are very, very little, very small. Okay, so this in a sense is the DNA replication elongation in eukaryotes. Now in this whole process I am telling about the replication fork movement and replication fork development and how they process the machinery, but I haven't focused on the chemistry behind the polymerization, and the chemistry behind two nucleotide joining is same for prokaryotes and eukaryotes, and everything, everywhere, in vivo, in vitro, same. We have 3 phosphate groups. Remember, the chemistry is very same. Let's say this is cytosine and we are joining it with thymine. This is say that the thymine is coming, and the cytosine that is previously present have a hydroxyl. The thymine that is now just there is having 3 phosphate groups, right, and a hydroxyl at the end, and this is the rest of the chain. This is the new nucleotide at the place. So what will happen? The existing nucleotide hydroxyl acts as a nucleophilic attack to the carbon, to the sugar carbon here, and what it does actually, this alpha phosphate is there and it releases the beta and gamma phosphate. So this attack to the alpha phosphate, sorry, it's not a carbon, this nucleophilic attack to the alpha phosphate removes the beta and gamma phosphate from there, and as a result what will happen? You will see the joining of C and T together, and what removes 2 phosphate groups are relieved. They are called as pyrophosphate or PPI. Now the idea is pyrophosphate, any molecule with multiple phosphate bond is energetic. So this pyrophosphate that we see here is energetic, and this pyrophosphate can be hydrolyzed, can be broken down into inorganic phosphates to get some energy, and the energy that we get by hydrolyzing the pyrophosphate into inorganic phosphates drive this reaction forward. This is the driving force of taking this reaction, because these are all enzymatic reactions. Remember, it should uplift that activation energy, and so that the reaction can process in the forward direction, and that is possible due to this hydrolysis of inorganic phosphate, the phosphate, this pyrophosphate to Pi, and then we get the energy to move forward, right? So that's the energetics part of this eukaryotic DNA replication elongation. That's why we move forward, right? So that's kind of it about the elongation. In the last lecture, in the next video, we will be talking about the termination process of eukaryotic DNA replication. So stay tuned and watch this series all through to get the understanding of DNA replication in eukaryotes. Thank you.